Okay, so today we are going to talk about the beauty of Lenti. Is why we want to use Lentivirus as a gene delivery tool, the advantages, biosafety, and protocols. So this is an overview. So first I will cover the why use Lenti as a gene delivery tool, and then so how the vector is designed and its biosafety. Then how Origin can offer the Lenti product to serve your Lenti needs. Then follows with the detailed protocols, step-by-step protocols. So what is lentivirus? So lenti means slow. So lentivirus is a slow replicating retrovirus. Most of the lenti vectors were derived from HIV-1, which is the enveloped virus, and the genome has 2 copies of positive RNA. Here are the viral genes. This shows the life cycle of a lentivirus. That's the normal wild-type lentivirus, not the lentivectors. So upon entering into host cells, the RNA genome is reverse transcribed into double-stranded DNA and then integrated into the host genome. The viral genome is transcribed and assembled into viral particles. So this is a summary slide showing the features of lentivectors as a gene delivery tool. It's high transduction efficiency, reaching to 90 to 100% in commonly used cells. It infects both dividing and non-dividing cells. Another important feature about lentivirus is it can stably integrate into the host genome, which is better for creating stable cells. So the cells listed here, primary cell, epithelial cell, neuronal cell, stem cells, they are known to be difficult to deliver gene by the traditional chemical-mediated transfection. So therefore, scientists have engineered lentivectors to deliver genes into those cells. Next, I want to show you some lenti transduction data. So this slide shows the lentivirus can infect hard-to-transfect and primary cells. The lenti-GFP virus were used to infect intestinal epithelial cells, mouse map cells, and primary vascular smooth muscle cells. So the gray pictures, those are phase contrast, showing the cells on the plate. And the green fluorescence pictures, that's the transduced cells. You can tell clearly that almost 100% of cells were infected, showing that lentivirus is a great gene delivery tool. And this data shows lentivirus can transduce ES cells, embryonic stem cells. The lenti-GFP virus were used to infect both mouse and human ES cells, and the pictures were taken 2 weeks after infection. The left panels were phase contrast showing the cells, and the GFP green cells, those were infected cells. So this data demonstrated that lentivirus is a powerful gene delivery tool for ES cells. And this is the in vivo transduction data. So lenti-GFP virus was injected intravenously into adult mice via blood vessels. And 1 week later, in both liver and spleen, you see very nice GFP expression. And even 3 months later, you still see very nice GFP, showing that lentivirus has been stably integrated into the host genome. So this data showing that lentivirus can be used in vivo in animal models. So now from the transduction data, you can see that lentivirus is a great gene delivery tool. So now, because lentivirus can infect human cells, you may have a concern. Is it safe to use? The most commonly used lentivector currently are second generation and third generation vector. This slide shows the second generation vector. So to make the lentivector safe, the scientists engineered the lenti system by splitting the replication essential, the viral replication essential, into different plasmids. So in the lentivector that carrying your gene of interest only contains the LTRs and the packaging signal psi, and the rest of other viral genes are provided on separate plasmids called packaging plasmids. So when you do a cold transfection of the lentivector with the packaging plasmid into cells, those packaging plasmids will provide the essential viral proteins to help this lentivector replicate and package into viral particles. But because the packaging plasmid don't have the LTRs and packaging signal, they will not be packaged. So that's why the particle called pseudoparticles. They are not the wild type particle. They don't have any viral gene in it. So only contains the lentivector, carry your gene of entry to be delivered into your cells. The third generation is even safer. The 5' LTR is a hybrid, so no need for no viral transactivity TAT. The 3' LTR is a thin LTR because it has a deletion in the U3 region. The thin stands for self-inactivating. This deletion can be transferred to the 5' end after integrating into the host cell. So that makes the virus unable to replicate. So the deletion in the 3' LTR makes the virus replication deficient. In addition, the third generation system, the packaging plasmid, are provided on 3 plasmids, further lower the chance of have the viral genes recombine back into the lentivector. So it makes the third generation is safer. So this is summary slides about steps using lentivectors. First, you make a pseudo lentiparticles, do a cold transfection with your lentivector with the packaging plasmid mix into HEC2NET 3 cells. And then 2 days later, you will have the pseudo particles make only contain your lentivector. And then you use your pseudo particle to transduce your cells. So these pseudo particles can still do 1 round of infection of your target cells, and it also can reverse transcribe and integrate into the host genome. However, due to lack of any viral genes and also the third generation, the deletion in the 3' LTR, there's no viral replication. Therefore, also no virus produced. So therefore, although the lentivirus can infect human cell, but no panic is necessary, as there's no virus produced in the after transducing the target cells. So what protocols need to follow for safety? So the biosafety level of lentivirus is biosafety level 2, which is the same as cell cultures. So you will need the personal protection equipment like lab coat and gloves. And the lentivirus need to be handled in tissue culture hood, also called biosafety cabinet. The biohazard waste decontamination treatment for liquid use bleach. And for solid waste, if you can pass through bleach and then autoclave, such as the pipettes, you pass through liquid bleach and then autoclave it. For details, you can follow the NIH guidelines. The take-home message is, if you can handle cell culture, you can handle lentivirus. So now I have shown you that Lentivector is a great gene delivery tool, and it's safe to use. So what does Origin offer to serve your lenti needs? Gene delivery, there are two types. One is the overexpression to deliver an seeding clone into cells, and another type is gene knockdown. You can deliver an shRNA into cells. And Origin offers both products, Lenti-ORF and Lenti-shRNA. The offer is comprehensive, it's genome-wide, and the species are human, mouse, and rat. For both products, there are two formats. One is plasmid, that we can have the ORF or shRNA clone into a lentivector, and then you will package in your lab. And another format is pre-titered, ready-to-use lentiviral particles. You can just receive the viral particles and add to your cells. The lenti accessories, of course, the packaging kit. You can use the lenti packaging kit to package lentivirus. And the control particles that serve as a negative control for your lenti transfection. Lenti concentrator, you can use to concentrate lentivirus to get higher viral titer. So this is a little comparison between lenti-plasmid versus the viral particles. For plasmid, as I mentioned, the first step, you need to get a lenti packaging kit and package the particles. So the average cost of a lenti packaging kit commercially is about $600, and it takes 4 to 6 days to do it. If you want to measure the viral titer, you need to get a titration kit. Commercially available, average cost is $600, and takes 1 to 7 days, depends on the method you use. In comparison, for particles, if you order it, Origin provides, as I mentioned, the pre-titer with the titer already measured. You can directly add to your cells. You don't need to do any of those steps, so you can save your time and money. Now, more details about Lenti-ORF clones. Any gene that's the same ORF is provided in 4 different lentiviral vector options with or without pure selection. So this is the lenti-CMYC-DDK vector. Your ORF will be cloned into these cloning sites, and it will be inframed with the MYC-DDK tag. So the protein will have the C-terminal MYC-DDK tag. You can use the tag antibody if there's no protein-specific antibody available. You can also use the tag to do affinity protein purification. So commonly, anti-DDK antibody beads or anti-flag antibody beads, DDK the same as flag, are used for protein purification. Origin also offers the anti-DDK resins. So this vector is a C-terminal GFP tagged vector. So you can, the protein will be inframed with a GFP. You can use GFP to monitor transfection or transduction, viral infection. And you can also track the cellular localization of the fusion protein too. So those are the 2 Lenti-ORF clones with the P2A Puro. So you can use Puro selection after transducing your cells. If you need more different tags or selection markers, Origin offers many lenti cloning vectors that either different fluorescent proteins or different selection markers, like neomycin, blycidin, and this vector even has is under EFY promoter. The ORF can be easily shuttled into any of those vectors. So lenti shRNA, this is a 5' LTR, this is a 3' LTR, and the sequence between the LTRs will be packaged into viral particles. So you can see there are 3 major functional elements within the LTR. Of course, the shRNA is under U6 promoter, and there's a puromycin resistant gene. There's also a GFP reporter. So therefore, in your transduced cells, you will express your target shRNA, and you can also use a Puro to select stable cells. your transduced cells will be green. Here the picture of our shRNA. It's a kit. It has 5 tubes, 4 gene-specific shRNAs. They have different genes individually in the tube. And this is a scramble control. It's negative control. So we offer performance guarantee of at least 70% or higher knockdown for our shRNA. So next I will show you some validation data. This is a CSF colony stimulating factor ORF clone in the GFP text vector. And the viral particles were packaged using Origin's packaging kit. You can see very high transduction efficiency. And you see strong, intense, the GFP expression showing the high gene expression too. The CSF is a transmembrane and a secreted protein, and you can see it's localized in the right cellular localization, suggesting that the GFP fuse the CSF going to the right cellular compartment. So this showing the Lenti-ORF expression of different sizes. From lane 1 to 6, you will see the ORF size ranging from 6.5 kb to 0.7 kb. And this is a Western blot with anti-DDK antibody. We see very nice expression for all the different sizes. And this data shows gene expression at different MOI. MOI stands for multiplicity of infection. That's the number of viral particles per cell. MOI 1 means there's only one virus per cell, and MOI 10 means there are 10 viral particles per cell. And you can see that the higher MOI is, the higher gene expression is. That means if you want to get higher gene expression, you can use a higher MOI. This is showing the effective knockdown using the Lenti shRNA. As I mentioned earlier that the Lenti shRNA vector between LTRs, it has GFP reporter. So all transduced cells will be green. And this is the scramble control shRNA. And this shRNA is targeting the GFP in the vector. So you can see significant knockdown of GFP. So now I have covered that Lenti is a great gene delivery tool and safe to use, and Origin offers great Lenti products. So what about the detailed protocols? Okay, the protocol 1 is Lenti packaging. So Origin's Lenti packaging kit is for the 3rd generation vector. The packaging kit has 3 plasmids and is pre-mixed, optimized at the optimal ratio for high packaging efficiency. So it's ready for co-transfect with your Lenti vector to produce high titer Lenti particles. So this is a diagram just showing that co-transfect your Lenti vector with the Lenti packaging plasmid into HEC293 cells. And then 2 days later, you will have the virus released to the supernatant and ready to transduce your cells. Quick summary, what you need. You will need the Lenti vector, either Lenti-ORF or shRNA, and Lenti packaging kit. So Origin's packaging kit contains the transfection reagent. If the packaging kit you don't have it, you also need the transfection reagent. And HEC293 T cells not supplied. So this is the Lenti packaging detailed protocols, step-by-step protocols. So D1, seed the HEC293 T cells. For 10 centimeter dish, seed about 2.5 million cells. And D2, do a cold transfection. In an Eppendorf tube, you add 500 microliter optimum media, and then you add 5 microgram Lenti vector and the 6 microgram packaging mix together, and then add 44 microliter Mectran. Mix and incubate at room temperature for 20 minutes, then add to your cells. And the next day, change the cell culture media. And D4, the virus is ready to be harvested. So you harvest the supernatant, do a brief spin to remove the cell debris, and then filter the supernatant with a 0.45 micron filter. The viruses are ready to use. For long-term storage, avoid repeating freeze and thaws. So aliquot the virus into small volumes and store at -80. If you want to measure viral particles, there are different ways to do it. Just follow the corresponding protocols. So the next procedure is transfection protocol. So day 1, seed your target cells. The confluence is about 30 to 50% confluence at the time of transfection. So different cell, the size is different, so maybe the cell number may be different too. On the 24 well plate for Hectona 3, you see about 50,000 cells. And at the day 2 transfection, first decide what MOI you want to use. So MOI, as I mentioned earlier, is a multiplicity of infection. That's the number of viral particles per cell. For example, if you want to use 10 MOI, and this is the total number of cells seeded in the well, that's the total units of virus you need per well. And this is assumed viral titer is 10 to the 7th units per ml. So that's total virus divided by the titer. This is the volume of virus you need to use per cell. So thaw the virus on ice, and then for the 24 well plate, per well you need to make a 500 microliter viral solution in growth media, so which contains 50 microliter of the virus. You also need to use a polybrain, which increases the transfection efficiency. So the working concentration is 8 microgram per ml. Then you add 2 cells. The next day, change the cell culture media. And day 5, 2 days later, that means 72 hours post-transfection, the cells are ready to analyze gene overexpression or gene knockdown. If your lenti contains any selection marker, you can directly split cells and add to the growth media. So I have shown you lenti system is a very powerful gene delivery tool for hard to transfect and primary cells in vitro and in animals. It is convenient and with a high transfection efficiency, and it's a simple protocol, and it's safe to use. So next, I will be happy to answer any questions. If there's any question not answered, you're welcome to email our PhD scientist at techsupport@origin.com.
Well, thank you, Dr. Liu, and I'd like to thank everyone for your attention. I hope you found it informative. This recorded webinar is available, as mentioned, through a link in a follow-up email that you'll be receiving, and also on Origin's webinar webpage. So now we'd love to answer any of the questions that you may wish to ask. And again, please do so by clicking on the question box in the control panel to your right. Do you need assistance?
Yeah, it's okay. Okay. All right. So one question is, all right, is it possible to clone an 8 kb ORF into lenti vector? Because the lenti genome is about 8 to 9 kb, and also you will have the promoters. We have tested to clone 6.5 kb ORF. I think the 8 kb will be too big. Okay, so one question says that because the packaging plasmid, why are the packaging plasmid are not in the pseudo particles? in the lentivector carrying your gene of interest has the LTRs and the packaging signal. The packaging viral genes, in those vectors, there's no LTRs, no packaging signal. For those viral genes to be able to package then it might need a couple of recombination back into the lentivector, which has the LTR. So normally, the packaging plasmid cannot be packaged because it does not have the LTR and the packaging signal. And one customer is asking if Origin offers CRISPR-Cas9 lentiviral packaging products. We have it. It's in the preparation of launching on the website. Okay. One customer asks, what are advantages and disadvantages of lenti AAV? I believe I do have the slide. Okay. I don't want to confuse two customers, so I did not show. So this is a comparison between different viral systems. And between the AAV, that's the genome is a lot smaller. It's only 4.5 kb. So because you also have in the vector, you will have promoters. You probably package 2 kb and less. So infect non-dividing cells. Both the lentivirus and AAV infect non-dividing cells. And the viral titer similar. With AAV, most episomal. It does not integrate into the host genome. Okay, one question says that the GFP control always have higher expression than the target, the gene of interest fused with the GFP. It is normal because the GFP is at the C-terminal tag. The gene expression of the fusion port depends on the expression level of your target gene. Depends on the gene. The translation efficiency and the protein stability is different. So therefore, different genes, the steady state level, the protein level will be different. Okay. Do you need to centrifuge to concentrate the lentivirus? It depends on your application. To work with the cell lines, our concentrator, you don't need to use ultra centrifuge. Yeah, just to use regular centrifuge. It's very fast and convenient. You can go to the website and check the protocol. It's very convenient to use, and you only need probably 2 to 3 hours to do that. So one question is, after lenti transduction, does it mean the cells can be selected at a stable cell line? The answer is, if the lentivector has a selection marker, such as our shRNA always has a pure selection marker, and the lenti-orf is also available in PureVector too, you can use the antibiotic to select your stable cells.
But the cells you select is a pool because the integration at different sites. So if they want colonies, they want cell line, which is the genome is pure. You need to.
Okay, if you want individual cell colonies, of course you can isolate a single colonies. You can just seed very at low confluence and let them grow into single colonies and pick the single colonies, and that's individual cell colonies. Or you can use it as a stable pool. There are two ways to do it. One question I believe is, how do you usually measure MOI for a batch of viral production? I think it could be mean that how do you measure viral titer for a batch of viral production? There are different ways. Sometimes they use P24 ELISA kits, and sometimes people use PCR. There are different ways, or they use infect cells to just calculate the colonies. There are different ways to do that. Here you mentioned MOI. Maybe you have other questions too. Different cell lines, the optimal MOI, you need to find out using the control particles to see which MOI give you a higher transduction efficiency, especially using the GFPs. You can directly see the gene expression level. One question is, do you have an antibody for GFP for verified transfection by Western blot? Yes, we do have a different GFP antibody on our website. If you have difficulty finding it, you can contact the tech support. Just let us know.
There's a customer seems to didn't get the point. Why should we use a third generation virus?
Okay. The third generation, because it's safer, because the 3' the deletion at the U3 region. So after integrating into the host genome, your target cell, the deletion just make the virus cannot replicate at all. Even if you have viral genes in the cell, it still cannot replicate because the deletion will be transferred to the 5' the LTR, and that's the promoter. That's the promoter for the virus to replicate. So it just will not replicate because there's a deletion in the 5' the promoter region. I'm talking too detailed, I guess.
Yeah, the key basically just to make the chance for viral replication even lower.
Correct. Yeah, it's safer, and it doesn't need for the third generation. It also doesn't need the viral transactivator TAT. For second generation, you do need that. So in the which will be provided in the packaging plasmids.
Here's a very practical question: how do you store the viral soup after harvesting them?
Okay. Yes. Because the virus is enveloped virus, to avoid the frequent freeze and thaws, you need to aliquot the viruses and in a smaller volume, and then freeze them at -80. Okay, one question is that, does the lentivirus integrate in random positions, or do the LTRs directly insertion in a specific spot? The answer is the lentivirus integrate into the genome randomly. So if you want to specifically integrate into a position or spot in the genome, the AAVS1 CRISPR-mediated AAVS1 transgene insertion, that's the way to go. It will specifically insert at the AAVS1 site in human cells. For detailed information, you can go to Origin's CRISPR website. There's a tab, AAVS1 insertion. Okay. Another question is a very detailed question. How many times do you harvest the supernate for the lentivirus? 1 time or 2 times? So you can harvest 2 times. The titer changes a little bit. The second time a little bit lower, but you can harvest both. Like the day 4, that's the first time, and you can add the new media and harvest again on day 5. Yeah, that's a following up question with the integration side with Lenti in cells. If integration is random, how does silencing of specific region genes work? Yeah, this is the problems with the random integration. For most research, it might be okay, but if you are concerned, please use the CRISPR-mediated AAV1 insertion. Oh, okay. So what is the question?
I think what we're talking about here is how is there silencing of specific genes if it's random integration? And I think I think the confusion is the introduction of shRNA.
Got it. Now I understand the question. Okay, the question asks if the lenti integration is random, how can the silencing, knocking down of a specific gene? Yes, the integration into the genome, then the shRNA will be expressed, and then the shRNA is gene specific, able to target the target gene mRNA, so the target gene will be silenced. Sorry, I didn't get the question first.
Okay, well, for all the other questions that we didn't get to, sorry, but we will follow up for sure with an email from us. So thank you for your time today, and thank you, Dr. Liu. Let us know what you thought about this survey that you'll be getting